completed my PhD in 2015. Then I went to Case Western Reserve Medical School in Ohio. I did my postdoc from there. Yeah, just a second. Yeah, I did my postdoc from Case Western Med School for nearly like 4 years there. So my PhD was mostly like organic chemistry. It's more of a medicinal chemistry where I synthesize, hundreds of molecules. Then I did screening against bacteria and cancer. Yeah. So, moving to Case Western Reserve, I joined in the genetics department where I did, hardcore biology projects there. So this is exactly my expertise is. Basically, I do both chemistry and biology. As the name of the lab states, it's chemical biology lab. All right. So I joined in IIT Kanpur in the Department of Chemistry in December 2018. It's nearly like 1.5 years since my joining. So here, I work in chemical biology where we do drug discovery and screening in different cellular disease phenotypes. So that's about me. And I knew Mr. Venkatesh from, I think it was back in 2007 when he came to NCL Pune for his master's project. And we have been friends since then, and he's an excellent friend. And thank you, Venki, for the invitation. All right, so let me get into the topic today. To give you brief knowledge about what we're going to discuss. So the presentation was prepared targeting students audience mainly. So there will be a lot of fundamental basic contents in the topic. And some of the experts who are teachers and researchers, please bear with me. And I hope like I'll be able to finish in an hour from now. Just to see like how it's done. All right. So in the overview of my talk, so first I will be talking about like viruses, and then we'll just move on to what is SARS novel coronavirus 2. And from then, like we'll see like how exactly this gets spread in human, like so fastly, and it creates like a very strong pandemic all through the world. Then we will see like what are the different diagnostic tools that have been developed in order to identify the SARS-CoV infection, SARS-CoV-2 infection particularly. And finally, we'll move on to like what are the steps that we have taken, measures that we have taken in terms of vaccine development. Where do we stand, and so what do we think like when do we think like we will get vaccine? So from then, I'll just give you a brief introduction about the virus evolution over this period. Basically, like but here the viruses mentioned are like really dangerous viruses. They are known to cause fatalities in human. So it is not like SARS-CoV-2 is really the new one, but back in 1931, there's a virus called the Rift Valley fever virus that emerged. That was the origin of the virus was found to be mosquitoes, and similarly Zika virus in 1947. Both of them came from mosquitoes. Then 1967, there was a virus originated from monkeys. They called like Marburg virus. And 1969 Lassa virus, 1976 Ebola virus. Even today we are still hearing about Ebola infections. So these three, particularly this Marburg virus, Lassa virus, and Ebola virus, these three are known to cause bleeding inside your organs. Basically, like the blood will be released inside your body without your knowing. So they are really, really dangerous. Ebola virus, for example, that causes like 98% fatalities. Means like once you get it, there's a very high chance that you die. So, and similarly, like 1994 Hendra virus, then Nipah virus, you all know, in 1990 it emerged, and recently it gave an outbreak in India, in Kerala. But Kerala government had done exceptionally well in containing the virus outbreak. In 2003 is when this extremely dangerous coronavirus identified, which is called SARS coronavirus, which is called the SARS-CoV-1. So what is SARS here is like we'll just see in detail in upcoming slides. In 2012, the other virus is called MERS, which is called the Middle East Respiratory Syndrome. As the name states, it emerged in Middle East. That was also found to emerge from that. If you see like overall in this picture itself, majority of these viruses, for example, starting from like Ebola, Nipah, SARS-CoV-1, MERS-CoV, and then this SARS-CoV-2, all of them originating from bat. So bat is really a strong host for majority of these dangerous viruses, and then they pass on to animals, not directly to human. And for example, Nipah virus was directly transferred to pigs, and then pig transfer the virus to humans. So that is exactly how the evolution process happened. SARS-CoV-1 was like found to transfer its genome to there's something called a civet cat that is known to exist in forest, and we have several in IIT Kanpur. So if there's a virus that is coming through that, and we have very high chances of ourselves getting infected. By the way, all right. So now this MERS virus, come from bat again, but that infect camels. As you know, Middle East is full of desert, and as you can expect, camels can easily get infected, and that to transmit the virus to human. In 2018, World Health Organization announced a nomenclature called disease X. So what is in the disease X is that if there is a disease where you do not know what is the root of cause, basically origin, but it causes very high pandemic and epidemic. So those diseases will be called as disease X. That's what the nomenclature has been proposed in 2018. And SARS-CoV-2 coronavirus was the very first disease X that has been identified. I mean, then later it was found that it is SARS virus, which is called novel coronavirus 2, and then the disease caused by the SARS-CoV-2 is called as COVID-19. So the COVID-19 is the disease name that is caused by SARS coronavirus 2. All right. So now let's get into what is coronavirus. And as a scientist, we always think about getting more into science than the superficial information. The scientific name of coronavirus is orthocoronavirinae. So, or coronavirinae. And if you look at the figure on the right side bottom, so this is the electron microscopic image of coronavirus. So if you can see the spikes of the coronavirus, the outside of the coronavirus almost looks like a crown. That is why the name corona came, which is in Latin called corona. So that's why the virus is called as coronavirus. And it was first discovered in 1930s, as the last slide mentioned. It was first discovered in chicken, basically. It came from mosquito to chickens, and there were several chickens dying with this virus due to bronchitis, that is your respiratory infection. Then human coronaviruses virus were first discovered in 1960s. They are enveloped positive sense single-stranded ribonucleic acid viruses. They are not DNA viruses, but they are RNA viruses. And there are 7 types of human coronaviruses known, and 4 of them are mild and cause cold, and also mild respiratory infections. And among them, the 4 are mentioned in here, where HCO, NL63229E, OC43, and HQ1. So these 4 coronaviruses are mild. And among these 2 are also classified as alpha coronaviruses and beta coronavirus. So what is this alpha beta? And not just only alpha beta. There are 4 different sets of these coronaviruses are classified: alpha, beta, gamma, and delta. So how this classification comes is that based on the genome sequencing. They all have pretty similar genome sequencing to be classified as coronaviruses. At the same time, there are differences within the coronavirus. So they classify according to the sequence identity as alpha, beta coronavirus. So these are the 4 viruses. As I said, they are known to cause mild infections. There are these other 3 really dangerous and highly infectious beta coronaviruses, and all of these 3 are beta coronaviruses. They cause acute respiratory diseases, so that the name severe or sudden acute respiratory syndrome, which is called as SARS. So these are all of them known to cause breathing difficulties, really heavy breathing difficulties. So like I said, what are these 3 coronaviruses? One of them, the first one is SARS-CoV-1, which was identified in 2003 in China, which led to 8,422 cases, and 926 were fatalities, which is nearly 11%. And only 3 cases were found in India, so we are fortunate in that respect. And then this Middle East respiratory syndrome, MERS, which causes 2,599 cases, where 866 deaths were recorded, which is 35% death rate. So that's quite heavy. But then came the SARS coronavirus 2 in 2019. I took this number this morning at 9:30, where 6.85 million people have been infected, and then 398,000 people are already passed out. So if you look at the percentage, definitely the SARS-CoV is much lower than MERS. But if you look at the volume of death, it's really, really heavy. So that gives you an alarming signal that we need to be really careful about getting it outside. Okay, so now couple of slides back, I mentioned something called enveloped positive sense single-stranded ribonucleic acid viruses. So this is a long sentence, but this has like several meanings. For example, enveloped, it is easy to understand. It is like a ball where the virus material is packaged into a ball. So that is why it's called envelope. And there is something called single-stranded ribonucleic acid that you all know. No, ribonucleic acids are single-stranded. That makes sense. So it's a RNA virus. What is something in positive sense in here? So to explain that, first of all, I need to give you a brief introduction. Like I said again, tell you this presentation was targeted towards the students. So there will be a lot of basic contents, contents I said. So this is about the central dogma of life, where DNA is converting into RNA and RNA into protein. How this exactly happens? So in our body, in our cells, like eukaryotic cells, we have chromosomes, and they are like double-stranded bDNA packaged into a ball-like structure, which is called the chromosomes. And the chromosomes taken up into a RNA machinery where the transcription is taking place. So basically, what happens is this DNA unwinds to form like a two single-stranded DNA, and then DNA biosynthesis starts, RNA biosynthesis starts with the help of an enzyme called RNA polymerase. So basically what happens is like this, already ATGC, you get the complementary pairs, like A pairs with the T and then G pairs with C. So in the case of RNA, your T is getting replaced by uracil, U. So now what happens is like you have the DNA sequence. This is called a template sequence. So the template sequence is read by in an enzyme called RNA polymerase. So this RNA polymerase comes and binds, and then this senses what is this nucleotide. For example, this is guanine G, and it brings C from outside. So this entire process is happening in the cytoplasm, that is inside the cell. All right. So now this is exactly how the RNA is synthesized. So we will keep on be adding, and wherever there is A in the place of DNA biosynthesis, it will be T, but this is RNA biosynthesis. That's why you get U in the place of A. So now you synthesize the mRNA, your cell basically. Now you means like your cell synthesizes the mRNA. This mRNA in eukaryotic system is called as pre-mRNA. What is going to be pre-mRNA is that this mRNA cannot directly turn into protein. It doesn't go directly into translational machinery where the proteins are synthesized, but instead it is processed in the cytoplasm. So this process happens in the cytoplasm, right? So what is the process that is exactly happening is that not all the entire sequence turn into protein. So there are some nucleic acid residues that is useless. So which is called a non-coding region. We do not need them for particular protein synthesis. So if you do not need for protein synthesis, why do we need? So that is why it's processed. The mechanism is called a splicing mechanism. I'm telling you a lot of words, just for you to like, get hold of those words so that like whenever you get to read, you understand better. All right. So this processing of pre-mRNA into mRNA mechanism is called the splicing mechanism. So the unwanted region in the RNA is cut down. So now this produces the mature RNA, right? This mature RNA goes into the ribosomal complex. In the ribosomal complex, you have protein, ribosome, and you have something called tRNA. So basically, tRNA is the RNA that has amino acids. So these have like three nucleotide residues. These are called codons because they code amino acids. The guy come and find a complementary sequence and load the amino acid. So now if this is not matching perfectly, it will go. Next amino acid will come. So we have for all the 20 amino acids, we have 64 codons existing. And out of those 64, 61 are the coding codons. And then there is something called a stop codon. There are like three. Basically what they do is like they basically signal the stop the protein synthesis. Wherever you want to stop you cannot like keep synthesizing the entire genome, right? Because every protein is like really smaller in size compared to the entire genome length. So you need to stop at certain point. They are called the stop codons. Stop codons will stop the protein synthesis. Okay, so now let me just to summarize this slide. We have chromosomal DNA that is getting transcribed into pre-mRNA. Pre-mRNA is processed into mRNA through splicing mechanism. This mRNA is called mature RNA, and that goes into translation for protein making. All right, so this is the entire mechanism happening in eukaryotic cells. So what happens in the coronavirus? What is positive sense is that, so these are RNA viruses. There is no point of thinking about transcription itself. So they can just directly go into translation process. So positive sense RNA directly translated into protein. So they are already mRNA. You don't need to do any processing. And there are viruses, for example, Ebola virus or Marburg virus. They are called negative sense viruses because they come up with an RNA only, but that is pre-mRNA. So that needs to be processed. So here, what is the processing is that they are complementary RNA. All right. So now you need RNA polymerase. RNA polymerase, what it does, like in the earlier slide we had seen, it goes and binds to the DNA. It synthesizes the complementary mRNA, right? So here, the RNA polymerase will come and bind to the RNA again, but it will synthesize complementary RNA. So that is what here I put in as like it does not undergo translation, but needs RNA polymerase enzyme for making complementary mRNA, and that will get converted into protein that is called translatable RNA, which is mRNA. So now I hope you guys understand what is positive sense and a negative sense. You know, in Tamil, there is a saying, the Vadiyalu says that, something like that. So which means like you just come directly and then start working on it. So this positive sense is basically means that. So you have the mRNA that is ready for translation. That is why they spread so faster. Okay, cool. Now we'll look at the components, right? Like, what are the viral components and how exactly these guys spread. So if you look at the virus component, I will start from the top right. There's something called membrane protein, which is called M. Basically, this membrane protein gives shape to the entire virus. So now this is really key for like protecting what are the material inside the virus. And there is something called the glycoprotein S, which is called the spike protein. And this protein is a key protein, really key protein for recognizing human cells. You see, like now the first sentence what I had written, it is called the spherical particles. So they are not lives; they are simply particle. Unless they get into a living system, like human, rat, or whatever, it doesn't get life at all. It's just a single particle. It just stays there and dies there. No issues. But once it gets a host, living host, it can multiply into several particles, millions and billions. So now this glycoprotein is the key protein that helps for getting inside the human cells. We'll just see the mechanism how exactly this guy happened at the more scientific level. And then there is this single-stranded RNA. If you look at that, all the single-stranded RNA is like present inside the particle, and the entire RNA is protected by a protein called nucleocapsid protein. So this protein protects the RNA from all the external environments because RNA is quite sensitive. So if somebody is asking you DNA is more stable or RNA, it's very easy to answer DNA. That is why DNA is our genetic material and not the RNA. Why RNA is unstable? As you know, like there is 2' hydroxyl group in the sugar backbone, and that can undergo cross reactivity with the phosphodiester bond, and it will form cyclic phosphates. So it's quite easy, and if there is any slightly basic environment, that is good enough to trigger the entire RNA collapsing. That is why RNA is unstable. That needs to be protected. So here in the virus, it's the same thing happening because it is not DNA, it is RNA, highly unstable. So this nucleocapsid protein protects the entire RNA. And there's a protein called envelope protein. So as you have seen earlier, the envelope protein is key for packaging this virus. So the virus particles like packaged like this, and the envelope protein is like really key for making all these proteins to be together and making the entire stuff. So this lipid bilayer membrane basically a part of this membrane protein, but that makes the composite where it forms like nice boundary where it is not hydrophilic, where it cannot dissolve. It's almost like a cell. So let us look at like how this coronavirus signals human cells such that the human cell is like allowing coronavirus. It is not that easy, like in human, like every mechanism, biological mechanism evolved with a specific roles. So it is not like just like that anybody can get inside. And so how cruel this virus is, and beautifully they get into the human cell. Let us look at the mechanisms, like detailed biological mechanism. So here is the paper which came in Science in 2020, like in April 2020. So here they beautifully explain what are the different proteins that is involved in letting the protein inside. So in this paper, they mentioned as 6 steps for the virus particle to get inside the human cells and then replicate into multiple numbers. So the first step is called attachment and entry. So in this, you see, like I said, this is the SARS-CoV-2 protein, and the green part is called the spike glycoprotein, S protein. So this S protein is binds like recognized by a receptor protein called ACE2, angiotensin converting enzyme 2, and this ACE2 is rich in human cells. Like it is why it is rich. It is not actually meant for recognizing virus, but it is meant for recognizing many small molecules in our body for like regular cells function. But this virus is really smart that basically like hijack the system, human system, like a cell system, such that this protein can also recognize virus proteins. So the spike protein is recognized by this ACE2. So these two comes and binds, as you can see it here. It's like strongly binds. And then there is a protein called TMPRSS2, transmembrane protease, serine protease. So what this does is that this basically fuses this virus with a human cell, something like this. So basically this cleaves the membrane such that this can beautifully enter inside. And once it enters, this entire virus is packaged. You see, it is like packaged. This is the virus, and there's a coating layer on the top of it. This mechanism is called the virus particle getting inside the human cell is called endocytosis. Endo means inside, cyto means cell, and osis means like basically breaking inside. So the process is called endocytosis. And we have the top of there's a membrane protection that is basically packaging the virus to get inside. Those are called endosomes. Basically, they act as transport vehicle. We call it as like cargo. Basically, it's like a lorry, transferring material from one place to other place. So it's basically taking the virus from outside and then taking it inside the human cells. Now, I'm going to introduce a term called drug repurposing. So I don't know how many of you here, particularly with students. That is something, one of the really strong research area, promising research area emerging. And for example, now like nearly 60 to 75% of the funding for drug discovery to target COVID-19 were given for drug repurposing. What is drug repurposing? Drug repurposing is basically there is a FDA approved drug that is commercially available and for treating some other diseases. For example, it can be antibacterial agent, it can be anti-cancer agent or anti-diabetic. So take those drugs and see if that drug can also kill this SARS coronavirus. So basically, you are already having a drug and repurposing into doing some other function. So this is called a drug repurposing. And here are the two drugs that the totally three, but these two are drugs, chemostat and nefamostat. And these two molecules have been approved by FDA for some other purpose. For example, chemostat is approved for I think it was like a cancer treatment, and nefamostat is for pancreatitis. So these two drugs are approved already in Japan, and they are currently being used. But what they found recently, after this SARS-CoV-2 emergence, these two molecules known to inhibit the protein TMPRSS2. So if it inhibits, what happens? The virus cannot get inside the cell. So basically, you are blocking at the entry point itself. And the other method is recombinant human ACE2 protein. Basically, you send more amount of this protein. So when you eat more amount of this protein, what will happen? Your cell protein, instead of your cell protein binding to virus, your external, what is the protein you are eating, that will bind to the virus. So means this will stop the virus from entering into human cells. So this is a step one. And in step two, it is called uncoating, where the endosomes basically breaks and releases the virus, and the virus again breaks out, and the viral DNA is released. This is called RNA. It is called genomic RNA. So that is released inside the cytoplasm. So now, already these two drugs are repurposed. Chloroquine and hydroxychloroquine, they are anti-malarial drugs, and azithromycin is antibacterial drug. And these, the drugs I'm showing right now are in clinical trials, by the way. So these drugs are known to block the viral genome release. So basically, like it blocks at this stage so that the virus cannot fit into multiple copies. All right. So the third step is called this genomic RNA replication. So in that particular step, like multiple copies of genomic RNA is made by a protein called RNA-dependent RNA polymerase. Okay, fine. So now there is a question, right? Earlier I said this just comes with only gRNA. It does not come with RNA polymerase. So now where do they get this RNA polymerase? So now what happens is that this sequence, genomic RNA, also has a sequence region for this RNA-dependent RNA polymerase synthesis. Your human cell synthesizes this protein, and then once this protein is synthesized, that basically acts on this and then makes more and more copies of genomic RNA. So now this RNA-dependent RNA polymerase also made inside the human cell from the viral genome, to be like precise. And then there is this translation process that's happening. The polypeptide chains, gene RNA are like come together to make like several polypeptides. for making more and more virus, and the transcription also takes place for making more and more RNA. And these two drugs, remdesivir and then flapiravir, are currently being used as inhibitors of RNA-dependent RNA polymerase to stop at this stage. At least, like, we can stop from the gRNA replication, genomic RNA replication process, so that we can block the viral multiplication at this stage. Cool. So now I said this final translation process that's happening. So this, in the gene replication process, you produce a lot of mRNAs, and all these mRNAs translated into non-structural proteins, structural coating proteins, and then many RNA. So now you have RNA, non-structural protein, and then structural coated proteins. So structural proteins are basically membrane proteins, envelope proteins, and then lipid bilayers. Non-structural proteins will be like inside, like nucleocapsid protein, and it is envelope proteins and all these proteins. So now you have all the components you need for making one more virus. That is what's happening. The fifth step is called assembly. So you bring the protein together, and the protein is synthesized in the endoplasmic reticulum of our cell, which is around the nucleus, and the Golgi apparatus that is getting translated into the Golgi apparatus for like further processing to make a perfect functional protein. Because endoplasmic reticulum may make the protein, and it's again like in Golgi apparatus proofread in case if there is a mistake, and that is corrected. So now the protein is ready, and the DNA is coming, your gRNA is coming from here, and the entire stuff is like getting packaged. And now the packaged fresh RNA. Now I'm just showing only one RNA. So one virus particle, one viral particle can make billions of particles through this mechanism. So now getting this virus exit outside is called exocytosis. So that is the sixth step is called virion release. Basically, that's active virus that is released from one cell. So one even just a single viral particle can get inside, can make multiple copies of virus. So this is exactly how the virus is spreading through our body through mechanisms. So like let's again like zoom out and see like how exactly this gets spread, particularly these coronaviruses. SARS-CoV-1, as we already said, like it is coming from bat, and then it has been transferred into this civet cat, and the civet cat transmits the virus into human. And similarly, MERS-CoV, that is actually giving the virus to camel, and it's coming. But we do not know exactly where the SARS-CoV is coming from. But people do say they see that it is coming from bat. But what is the second element that transfers this virus from bat to human that we do not know? And there is a recent protein sequencing information that they find the sea turtles are the genome is matching with the sea turtles virus viral infection. So it is spread in the Wuhan city in China, where in the fish market, basically that's where people are suspecting. So it kind of makes sense. It could be potentially from sea turtle. We do not know right now. All right. So let us now we understand like how exactly the virus can get in, and so what it can do once it gets inside. So it can like really cause a lot of symptoms, and 88% of the cases known to have fever, and mucus protection 33. You get like a dry cough 68%, shortness of breath. You see, there's nothing like 100% symptoms. I mean, there will be somebody, you cannot just say that like, no, I have only fever, I don't have cough, so I'm fine. Because the rest of the 12% do not have fever. So you need to be careful. And similarly, like, when you have mucus protection, it's only like 33%. You don't have cold, it doesn't mean you do not have COVID. I'm not scaring anybody. I'm just saying be precautious. Like, yeah. All right. So similarly for sore throat and muscle pain, when you have like multiples of this, and then you need to be really careful. You should be like isolating yourself from the rest of the people. Okay. So now there is the problem. The problem is peak viral shedding is at 24 hours prior to symptoms onset. Basically, you do not show fever, strong fever. You do not show like in cough or sore throat, any of them. But when you show 24 hours before itself, you have very high loads of virus. Means like you are highly prone to spread those virus 24 hours before itself. So by the time you knew that you suspect, not knew, you suspect you might have a COVID-19, you might have spread already to many people. And similarly, the viral loads in upper airway decreases significantly after like 2 to 5 days. So generally like after 4th day, if you see, it is nearly like 60% of the virus levels low. So let's say like you had infection today and you did not go to the hospital for like 5 days, and then they tested after like the 5th day, they may not even detect that you have COVID-19, by the way. And it doesn't mean you do not have, so you need to be careful about it. So these are the two information that I want to share. And here is the data from Nature Medicine that came like a month back to show you that how serious the case is. So if you look at the y-axis, it's called the CTA value. The CTA value basically means it's called so in a crude term, the amount of RNA present. Okay, in the virus. So means like basically it's going to tell you what is the level of protein on a virus which is present. Higher, I mean 35 means like very high loads of virus, and then lower means like low loads of virus. So at least 45 is the cutoff they have. So that's what you need to know about this graph. And you see, like, it was a case study that is done. And there was a woman who visited China, Wuhan, and then she arrived in Australia like 11 days before. So first 11 days she did not have anything, and she had on the 0th day she had a little bit of cough and a little bit of fever. So then she went to the hospital, and she said, no, expressed that no, I have a little bit of cough and fever, so I do not know what to do. And they started like doing the viral loading screen, and they found that on the 4th day you see the highest level of virus you see, but only on the 5th day you again get some cough and some fever. And when it goes, you see the green signal that okay. So now I'll just tell you like few things. So this high loads are seen like on the left right side. I have like a few more notations. I will tell you what it is, but no, let's just look at only the graph. So on the 6th day it goes down. By the time 7th day you'd almost see nothing. Means like you won't be able to people won't be able to diagnose whether you have COVID-19 or not, because the viral load is like really low. So in case if you have those symptoms and you want to really care about your family, you need to go immediately to make sure that it doesn't really get into other people. So now if you see like on the right side, I said like we'll just see a few more stuff. In the throat, swab, like and whole blood, urine, rectal swab, all those things are being taken, and where you do not see like much. But from nasopharyngeal swab, you take the viral swab so through your nose. When you take through your nose, you get like maximum amount of viral loading. So that is the preferred method of testing. And sputum is basically the saliva that is from your lungs. Basically, you cough and spit. And from that also they see like significant amount of virus, but on the day 6, like by the time, by the, you can somebody do the muting. I'm getting a lot of background noise. I'm sorry. Thank you. Yeah, all right. So now you see like on the 6th day, they see like very high viral load, but that is in sputum, means that is already from the lungs. So you won't be able to diagnose anything through nasopharyngeal, not anything. Really low amount of virus only you will see. On the 6th day, if you're doing like this kind of a swab methodology, you need to measure it from sputum and feces. People do take like these two samples. Quite often they do the testing. All right. So now it is so important. You need to maintain social distancing, and wearing mask are absolutely essential. Otherwise, like you will not know like when you will get infected. All right. Now let us look at like now we know. So there's this COVID infection. How are you diagnosing, and where are we going right now, and what are the methods that have been developed? So I'm just showing here the overall like what are the different methods that have been published so far and commercially being used. There's something called antibody-based immunoassay technology, and we are going to see what it is. I'm going to briefly explain about that. Basically, you add virus, and then there's the antibody that signals that your SARS-CoV-2 is present. Second thing is you isolate RNA, extract RNA from your virus, and you do a whole genome sequencing. You do the entire genome sequence. That's highly expensive, and it's not possible to do it like in a large scale. And similarly, like you can convert RNA into DNA. Like in the transcription process, DNA can be converted into RNA. RNA also can be converted into DNA. That is called a reverse transcription. Basically, you can convert it into DNA. You can make it into more stable genome. Now, RNA is like unstable genome, right? You can convert into cDNA. So now this becomes more stable genome. And again, do the genome sequencing. It's not viable, like I said, for the larger scale. And there's something called the real-time RT-PCR. That is like commercially being used by many countries, and that's been quite positive and then useful in identifying several cases. Of course, like there are issues. I'm not saying it's perfect, but definitely that is the technology, more reliable technology we have currently. And then there's something called nanopore target sequencing technology, and this has been like developed based on RT-PCR using the cDNA again. And there's a cool technology that came called ShareLab. I'm going to explain what ShareLab technology as well. So where you take the RNA and you can identify if there is any code. This is one of the most sensitive techniques actually, but it's quite expensive. So that's what people are worried about, like commercializing it. People are still working on it anyway. And there's something called PCR amplification, paper-based biomolecular assays, where you can drop this PCR product, which is basically RNA, and see like you can see signals. But this is more of a qualitative assay than quantitative. So this is not like pursued like really strongly. Cool. Again, this is for the student audience. I'm going to tell what is RT-PCR. So this is introduced in 1977. The PCR means polymerase chain reaction. RT means reverse transcription. So basically, you convert RNA into DNA. That's reverse transcription. And the polymerase chain reaction is making more and more copies of this DNA through this chain reaction. So this is introduced in 1977. And this RT-PCR basically allows you to measure the lowest quantity possible in DNA. For example, in forensic department, it's like very strongly used. You just identify one small hair and you'll be able to track who's the person is. So the technology that is used in there is like PCR, RT-PCR for identifying your gene. And in microbiology, this is one of the essential tools, and where for gene expression analysis, cancer research, drug research, forensic analysis, everywhere people use this technology. And even including like disease diagnosis and management, viral quantification, food testing, and there's something called percent GMO food. What is GMO? It is genetically modified organisms. Basically, let's say you are exporting a food from India to China or India to US. They do test the food for genetic modification. If there is any genetic modification that is bad for health. So even for that, PCR is being used. Animal and plant breeding and gene copy number for everything. This PCR is used, and this is the machine that basically multiplies the copy, and this is the machine that is known to read the output. We will see how we will read it. So now let us the practical experiment, how exactly we do. So now you have the swab, it's almost like your earbud, and insert into your nose, you get the sample, and store it into a buffer where the RNA or the entire virus is stable, because during the transport, if the virus is unstable, it will completely die and you won't be able to detect. So there are like specific buffers developed to keep the virus alive, or like alive means like stable, because it's not a living system, by the way, it's a particle. So to keep the virus particle stable, then you move into a buffer where the virus can be lysed. So here is the virus will all be there, and do the centrifugation, and you can extract the genomic RNA from the virus particle. So now this virus genome is like extracted, and there's a column you pass through it, and only virus genome can be isolated, and rest of the molecules can be thrown out. So now you have the virus genome, and I'm going to explain like some of the steps in later slides as well. So, but you just get the overall picture about like how RT-PCR works for COVID-19 testing. So this is the RNA, genomic RNA, and the green ones are like complementary DNA. So in this particular step, what you do is that you make into complementary DNA through reverse transcription process. All right. So you add this reverse transcriptase enzyme and add into the instrument that I showed earlier, polymerase chain reaction instrument, PCR we call it, and then that will make multiple copies of this cDNA. So once cDNA goes inside, like exactly like in DNA synthesis, biosynthesis, or RNA biosynthesis inside our body. So what are the components that we need for DNA or RNA biosynthesis? We need all those nucleotides, ATGC or ATGU, those are not AT, AUGC. So those nucleotides you need, and you need a primer. So what is primer? Primer is a starting material. Means like in cells it's easy because there is this RNA polymerase and there are other complexes that basically gels together to signal where to start the DNA synthesis. But here you are doing yourself, so you need the starting point, where to start. So for that, there's a primer. So basically you synthesize a short nucleotide sequence that is typically 7 to 8 nucleotide or 22 nucleotide lengths, and you put that, it goes and binds to complementary sequence. So what is complementary sequence that's going to bind? The primer is basically complementary sequence to the genomic RNA, which means you need to know the genomic RNA sequence as well. That is why the first thing what they do, any disease, first they do the whole genome sequencing, and thanks to the development. So now you know the whole genome sequences and you know the length of the genome. And now what happens? You synthesize the primer that is complementary to that. Anywhere, not necessarily like in one particular place. Anywhere you synthesize, you throw it, it will go and bind, and it will synthesize the DNA from that particular nucleotide. So now once you make like more and more copies of the nucleotide, so what happens? This primer has something called a fluorophore. So I will just explain this step, like how it is making more and more copies, and what is the result that you read in the next slide. Cool. So now I said there is reverse transcription. What basically reverse transcription means, you can convert RNA into DNA. Again, for this, you just need a reverse transcriptase enzyme. Also a primer. Where to start? The primer goes and binds to the genomic RNA, and from here, the synthesis starts. Again, like, so another complementary strand, it can again get synthesized. So many DNA can be amplified. You can make one DNA into several DNA. That's exactly how PCR works. All right. So now, if you want to do the PCR, like you want to synthesize more and more copies, you need energy, right? For example, in our body, there's so many ATP molecules synthesized, adenosine triphosphate, that serves as the energy source, and that gives the energy for DNA synthesis. But in the while, like, how do you give energy? Energy is basically the temperature. You heat it, and you are giving energy in the form of heat, so it can do. But majority of the proteins and DNA and RNA, these guys are quite sensitive to higher temperatures. How exactly, particularly proteins, even RNA, DNA, you can stabilize it for some time in heat. RNA is quite sensitive, but DNA is a lot more stable. You can even at like you can see 95-degree heat, nothing happens to the DNA. So, but now the protein, whatever the polymerase protein you have for synthesizing, that is not stable. So then people have isolated a protein from thermophilic bacteria. There are bacteria that is known to live at temperatures more than 120, 140. There is something called a thermus aqueous. So this bacteria is known to have this DNA polymerase that is surviving even at very high temperature. So they isolated the protein, and that was used here. So that is like doing the protein synthesis. That's called a Taq polymerase. So now that can synthesize. Basically, you heat at 95 degrees, and this becomes like single strand. DNA synthesis, it needs to be single stranded. So this is called a denaturation process. You make into two individual single strands. There is something called unhealing. There is something called unhealing. So what happens is that you cool at 50 degree. 50 degree, the temperature lowering is not good enough to form again double strand, but it is good enough to form the primer to bind. So now the primer is bound. Now you have the protein already, RNA polymerase. It will just go on to do the protein synthesis. DNA synthesis. So it's going to synthesize several copies, and you will get multiple copies. And here is the practical view of how exactly we set in the instrument. So what we do, we start with 50 degree, and we will ask the machine, we set the temperature to 95, and we ask them from 0 minute to 2 minute you stay at 95. So that is good enough to do the denaturation process. Then again, we do it slowly, adding it's called getting the temperature to lower for primer unhealing. Primer joining. And we ask them to wait there for 1.5 minutes, between 2.5 minutes to 4 minutes. And then again increase the temperature. This 72 degree is when the DNA synthesis is really the most. So let's say you have 100 copies, one copy multiply into a 2. So now it will become 200 copies. Now you have 200 copies. That's done. So what do we do? Now it's all double stranded DNA. And again, what you do? You heat at 95. It makes single stranded. Bring it to 72. Make more copies. You just keep on repeating this cycle. So you can just keep multiplying this protein. And here in this and the next step. So once you multiply so many number of copies, how are you going to detect? There are two ways of detecting. One is called cyber green, and that is a fluorescent dye. So when there is more number of DNA, when it binds to DNA, it becomes fluorescent, highly bright fluorescence. So when there's more amount of DNA, then it will be highly fluorescent. When there's less, it will be low fluorescent. So you can see if there's no genomic DNA, it won't be colorful. And if you are COVID-19 positive, there is genomic DNA, and it will make more copies, and you'll be it will be glowing really high. If it's negative, you won't see that. Simple. But this needs more DNA to be synthesized and slightly less sensitive. But there's something called a TAC-MINE. So what I said, there's a primer, right? The DNA synthesis from there. And along with primer, they also put one more primer. This primer is not for starting, but going and binding in between. So this has two chemical tags attached to it. One is fluorophore, another one is quencher. And this naming is called fluorescence resonance energy transfer. One absorbs at one wavelength, another one absorbs at another wavelength. But the first one, which absorbs at one wavelength, emits a light, and that emitting wavelength is absorbance for the other one. Basically, it transfers the energy from one absorbance to another molecule, and that absorbs that energy. It doesn't lead to the dissociation. So now what happens? If this is the fluorophore, this is a quencher. So for fluorescence, what you need to have, basically, the molecule should go to the excited state, and when the molecule decays to the ground state, it needs to emit in the form of radiative decay. All right. So when it's radiating, then you will see the fluorescence. But what happens? It doesn't allow the radiative decay, but it takes up the energy. Other mighty. So means no fluorescence. But the moment you cleave these two, then fluorophore is separate, quencher is separate. There won't be quenching, means the entire thing becomes fluorescence. So here, while synthesizing itself, it will keep synthesizing on this direction, and there is a block. So this will be cleaved at this place. RNA polymerase will cleave this guy and again keep synthesizing. The more number of atoms, the primer it cleaves, more fluorescence, you'll get more and more signal. This is a lot for RT-PCR. I hope you guys understand. And then let's just move on to the calculation. So now this is a busy slide I prepared for my class students. I'm just showing you but I will tell you in brief. So this is something called exponential increase in number of DNA biosynthesis. Basically what happens is that when you have one DNA and it becomes two and two becomes four, so four becomes multiple copies. So it is forming 2 to the power of n number of copies. And typically in PCR, how many cycles are done? 30 cycles, 30. So you start from a number and then you continue for 30 cycles. So 2 to the power of n number of copies you can make. No, in chemistry, we see that number in stereochemistry, where if you have n number of chiral centers, you can make 2 to the power of n number of stereogenic molecules. Anyway, so now it is similar to that, where 2 to the power of n number of molecules, you can the copies of genomic DNA can be made. You just imagine, let's say, even at a 22nd cycle, you have 125,000 copies. Just imagine 23, it become double, 250,000. And 24, it is 500,000. By that time, 25, you have 1 million copies. You just imagine, just 22nd cycle, you have 125,000. But in 3 cycles down, 4 cycles down, in the 4th cycle, you have 1 million copies. And I keep doing this until the 30th cycle. So let's say you have very little of DNA, you can make a huge amount of DNA out of this technology, RT-PCR. That is why this method is so reliable and it works really well. So to summarize this RT-PCR, basically you take a nasal swab and then you extract this mRNA and do the reverse transcription, get the DNA and add your primer, nucleotide, DNA binding enzyme and fluorescent dye, and then do the RT-PCR in the machine, and you'll make multiple copies, and you can measure the fluorescence using the fluorophore whatever you are having. And positive will have very high fluorescence, negative will have no fluorescence. That's all. So this is about COVID-19 detection using PCR. All right. So now there is something called Sherlock technology. I'm going to talk about. This is one of the very recent technologies that has come. People are expecting Nobel Prize for this technology, CRISPR-Cas9 system, and they keep evolving this process and bringing up high-end technologies. So what exactly? Excuse me. Yeah. So what exactly this CRISPR-Cas9 system is? Here it is called Cas13A. So I think this is really a big experiment, but I will try to tell you in short. So in CRISPR, what happens is that, like in the primer goes and binds to the genomic DNA. And here we have, instead of primer, there's something called a direct repeat sequence. And this repeat sequence will bind to the genomic DNA. So this is called the guide RNA or the primer, exactly. Like the primer in PCR, here this is the primer in CRISPR system. So this goes and binds. But this has this black thing. This has this signaling molecule. And this molecule binds to an enzyme called Cas13A. What is Cas? Is CRISPR associated protein. And this Cas13A will recognize only this loop structure. So it will come and recognize the loop. And what is this Cas13 is that is a nuclease enzyme. Basically, wherever you find this guy, it will cleave the DNA immediately. DNA or RNA, whatever, it will immediately cleave. So the single stranded DNA, RNA will be completely cleaved if the Cas13A is binding. If there is no genomic DNA, this will not bind. So there's no cleaving. If there is DNA, and this will go and bind, and this will RNA will be cleaved. So this is a therapeutic technology currently being employed. That's exactly how CRISPR works. All right. So now this in the Sherlock methodology is a specific high sensitive enzymatic reporter unlocking. So they call it as Sherlock. So in this technology, the additional component they add is a RNA sensor. Like I said, it's called the FRET sensor, faster fluorescence resonance energy transfer sensor, where you have fluorophore and the quencher. When it is together, there will be no fluorescence, but if it is cleaved, there will be fluorescence. All right. So now this sequence is again a primer. So what happens? Wherever this complex is there, this guy will also go. So now Cas13A, sorry, in this case, will not only cleave the genomic DNA, but also cleaves the sensor. So when this is cleaved, you get very high copy numbers of fluorescence. So using this technology, you can detect it 99% of positive cases. So it's really cool technology that has been developed, but it's very expensive. Like Cas13A expression is expensive, and this DR sequence synthesis is expensive, and you need to have this since multiple components and highly sophisticated techniques, so it's quite expensive. Yeah. So then we will just see antibody-based immunoassays. This is again like I will want to show you some basic information about like how things are working, but this will also give me a way to introduce vaccines as well. All right. So this is the platform that you have, whatever I have shown. So this is the platform, and in that you have something called secondary anti-SARS-CoV-2 N protein. So what is N protein? Nucleocapsid protein. So that is rich in coronavirus, and you cleave, you get the nucleovirus protein. So this secondary antibody is a protein that is known to recognize primary antibody of this N protein. So here I have this anti-SARS-CoV-2 protein, and this is primary antibody. And the primary antibody is tagged to European nanoparticle. Why European nanoparticle? European nanoparticle has like very strong excitation emission, and it is really strongly fluorescent, and you will be able to detect the presence of virus so easily. So now you take the SARS-CoV-2 virus, add a lysis buffer that will lyse the virus, and then just to tilt it, the virus will flow through this, okay, through the platform. When it flows, here is an antibody. This antibody is known to bind to the N protein, which is nucleocapsid protein. So if it binds, then this will also move, not moving. And this secondary antibody. So basically, how it's binding is it's a strong non-covalent interaction. So once it binds, it is very difficult to remove them. They are like really, really strong. So once this is bind, this is strongly binds, and then this flows towards this direction. And here is the secondary antibody. This secondary antibody binds to this primary antibody. Now this guy will get stick if there's nucleocapsid protein. But if there's no nucleocapsid protein, it just rolls off. Like when you're washing, it just like it keeps rolling out. All right. So now when you keep doing this, if there is virus, and you will see this is bound to here, and when you excite, you will see fluorescence. Understand? And this also has immunoglobulin antibodies. Immunoglobulins is rich in our blood. So now if this definitely, all of them will definitely bind to this guy. All of them, because they also have immunoglobulins, and we have immunoglobulin here, it will bind, and all of them will show. This is almost like, I don't know how many of you have seen a pregnancy test kit. There will be two lines. One is called a reference line, another one is called the test line. So in the reference line, everything will be lighting up. And in the test line, if it's not lighting up, which means negative. If it is lighting up, which means positive. So this lighting will tell you the assay is working. The kit is not damaged. The protein is not damaged, and everything is working. And both are positive means you have positive COVID-19 infection. So this is about the antibody testing. So, and this South Korean company, Celltrions, came up with this technology basically. Excuse me. Yeah, so basically they had a chip, exactly like I said, they labeled with antibodies for capturing, and they pass the sample through this, and they identify it is like exactly like the pregnancy test kit, two lines, if both are positive, and you'll get it. The excellent stuff about this particular technology is that in 20 minutes you'll be able to know whether you have a case or not. So this is currently being used in the airports of like South Korea, majority of the airports, because they just want to know like quickly whether somebody has COVID-19. So they are doing it. It's quite like effective technology, again expensive technology. So now I'm going to like slightly move to fundamentals. So immune system is our savior. Basically, it protects from all the diseases that comes to us, like any pathogens, hitting us, all those things. And so that basically saves us from diseases. There are two types of immune system. One is called innate immune system. Basically, you are born with this. Your cells have that immune system. So as soon as you're born, like you might have like different attacks, but nothing really is going to cause, because we already have the immune system. There is something called adaptive immune system. In the adaptive immune system, what happens is that whenever you get an infection, then the antibody produces. Your immune system produces a targeting protein that will go and kill. So this is called adaptive immune system. All right. So now this, I mean, the figure is like too big. I don't want you to see all these figures. What I want you to see is like there are two cell types from the stem cell. From the stem cell, like majority of the cell types that are being produced, and majority of them are like the immune system cells. For example, natural killer cells, macrophages, T cells, and B cells. All of them are like immune system cells. So now in the lymphoid progenitor, it makes two different cell types. One is called a T cell, another one is called B cell. That is coming from stem cell. We all originate from single cell, that is stem cell. So that converts into multiple different cell type. So few convert into like eyes, and few convert into nose, few convert into mouth. Like that's exactly how we entire human body is made. So the transformation is called a differentiation of stem cells. All right. So now here I have like some information about like what macrophages does. Basically, they take up all the bacteria and virus and kill them. and B lymphocytes. They are called B cells, I said, and they are known to produce antibodies. So in our body, antibodies produced by these B cells, and T cells basically helper cells. We will see in the next slide. And the cytotoxic killer cells, basically they are helping in killing some of the antigens that is coming in. Helper T cells also help in production of antibody. So here is the basic B cell function that we have right now. All right. So now the B cell, as soon as it has the protein, the recognizing protein, right? There will be one protein hardly. So the moment there is a particle that is coming in, it cannot immediately bind. So this B cell receptor is there, this is there, and the antigen is there, but there's no immediate recognition that's happening. There is something called helper T cell that is also coming from the stem cell that changes the conformation of the entire B cell, or does kind of a chemical signaling such that you recognize this. There's a new guy. He is not from our body. He is from external body. So this signals codes this cell. Now these two are recognized. The moment this guy recognizes, this B cell converts into plasma cell. So now this is the maturation process, differentiation process. Once this is differentiated, this makes more and more and more copy of this antibodies. So this antibody will be released inside the blood. So once it is really released inside the blood, what will happen? Any bacteria or like any pathogen, infectious pathogen getting in, these antibodies will go and bind to them, and then it will lead to killing of those pathogens. So this is exactly how antibodies produced inside our body, and then delivered for protecting ourselves from majority of the infectious pathogens. So fine, okay, our body is producing, that's all right. How do you know that this antibody is produced, and how are we going to test it? So it is not possible that they will infect you all the time and then generate the antibody to do the testing. So we need another method. They used mice. So in mice, what they do is they take the spleen cells where the majority of the antibodies produced, immunoglobulin antibodies, and they take the spleen cells, and there is this myeloma cell. This myeloma cell is what the B cells basically that is grown multiple fractions. This myeloma cells will not grow in certain conditions, specific conditions. So now what they do, they mix these two. In differentiation process, what happens? One cell can become another cell, right? So they give conditions such that these two fuse together, form a new cell type called hybridoma cell type. In this new media, only hybridoma can live, neither this live nor that live. So you get a new cell that is capable of producing more and more antibody, but also can multiply into several number of copies. So now you make more and more copies of this antibody, and that can be used for testing. So all those earlier testing that I have done, you need to load the antibody. That antibody actually come from this kind of synthesis. All right. So now to give you a overview about this antibody, if a person has an infected, their blood contains the antibody to the virus. Basically, it naturally produces that antibody that is binding to the protein, the viral protein. So that's called antibody. And there are immunoglobulin antibodies, and then immunoglobulin M and G antibodies. So few are short-lived and few are longer lived. So you need to synthesize the protein accordingly. Second, mixing of the blood sample with a known antigen. You just mix them, and this antibody linked to the enzyme. You just add them together. You have antigen, you add, and then you put a substrate. Basically, this enzyme substrate. What happens? This enzyme is also capable of reacting with fluorophores. So what you do when it reacts with fluorophore, like the FRET, exactly like the FRET, it will cleave the fluorophore, and you will see fluorescence coming. So that is what's happening. So once this is bound and the substrate is cleaved and then released into fluorophore, and if it is fluorescing, it is positive testing. So now the problem is you cannot just do it immediately. There are other issues in there. Okay. Now what you understood is how do we synthesize antibody? Antibody is nothing but vaccine. So that is why I introduced those concepts. So this antibody can serve you as a vaccine. You develop the antibody for a particular virus and take that antibody and insert into you, and it can basically protect you from the virus. So that's exactly how it's being developed in there. So now here actually I'm showing you the timeline of vaccine production. Basically, initial research takes up to 10 years sometimes, for the phase 1 trial itself. Preclinical testing research itself takes nearly 6 to 7 years. Then the preclinical development comes. That goes up to another 2-3 years, which is 13-14 years. Then there's clinical development. Clinical development basically means that preclinical to until the end, registering, marketing, and surveillance. And there's transfer process. So once definitely this vaccine can work against this particular disease, then you do the transfer. You take it into industries and ask them to make tons of copies for general use. That takes another 5-6 years. So that is why the vaccine development is not that easy. The development cycle is really, really longer. And now people are working really hard to find out the vaccine so that we can all get away from this SARS-CoV-2 infection. Cool. So now where are we standing right now in terms of developing vaccines for SARS-CoV-2? So as you know, here is the protein that we have already seen. There's animal protein, spike protein, matrix protein, membrane protein, nucleocapsid, and all those things. So now what people have done it they isolated the individual component and injected into mice. So what will happen when I isolate this only this protein and put it into mice? Mice will develop antibody for this particular protein only. Similarly, nucleic acid. Similarly, membrane protein. They tried multiple different methods, and they did they just directly taken the RNA and injected, and to see if they can produce RNA vaccine, and do the cDNA synthesis, inject the cDNA, and see if we can produce a vaccine. And similarly, recombinant protein vaccines. Similarly, there are several different techniques that have been employed, and they are in multiple different phases. So, but how long it is going to take? 6 months to 1 year, or maybe more than that, we don't know exactly right now. But we have a lot of promising research that is coming to the air right now. All right. So here are the some of list of vaccines that has been developed for SARS-CoV-2, by the way. And there is a group from Melbourne University. They have a vaccine that is in phase 2, phase 3 clinical trials. Though that shows really strong promise that can come anytime into the market. That's what at least we are expecting. And so here are some of the other vaccines from various different companies and research institutes. And there is a okay. So totally there are 28 vaccines currently. To give you the number, 28 vaccines are in the market right now. Market means in the pre-clinical and late clinical studies currently. And there's something from Oxford University. Their vaccine news came. You can see the date, 22nd May, 10 days back, or 14 days back, that Oxford University developed a vaccine, and that is in phase 2, phase 3 clinical trials. They already gave it to 300 humans, and it is showing really for positive feedback. So, and when we are at the edge of identifying a potential vaccine for COVID-19. So, and apart from Oxford University, this Pfizer company, it's a pharmaceutical company. They say that they have a drug for coronavirus, but that is in preclinical studying again. So they are still working on it. It's been more than 1.5 months. The news hasn't come out, so we don't know where it exactly stands. And 4 or 5 days before the Oxford University announced, the Moderna from US, they also announced that their vaccine is also in phase 2, phase 3 clinical trials. That also could come out anytime. So this is where we stand in terms of COVID-19 spread and all those things. And now I hope I gave a good amount of scientific information for you to understand how this coronavirus basically spread, and then what are the technologies that we have for diagnosis, and in terms of vaccine development, where exactly we do stand. And thank you for the opportunity and attending. And I'd be happy to take up any questions that you have. And Mr. Venkat actually suggested that we use the chat box for raising your questions. So you please write in there, and then I will answer it from there. Thank you.
Thank you, Dharma, for a nice presentation. Probably we can wait for 2 more minutes if we receive any questions from audience. Otherwise, we can wind it up.
Sounds good.
Thank you. Yeah, there is one more question.
Virus can able to attack bacteria. Yes. So what we do is if you want to make many copies of viral DNA before PCR came, guys, actually, I don't know how to track those questions because the questions keep coming. Then I will be missing. Apu, Venki, in case if I miss any question, you please let me know.
Yeah, sure. Yeah, sure.
All right. So to that question, before this PCR came, people use bacteria to amplify number of copies of viral DNA. So what they do is they basically let the virus get inside the bacteria through many different methodology. There's something called electroporation. So basically you apply short pulses of electricity, and the bacterial membrane opens up, and the virus can get inside. And once it gets inside, bacteria doesn't have nucleus; it's only cytoplasm. And in there, like virus gets in, and then the DNA is getting multiplied. Virus can attack bacteria. Yes. How long will it take for producing vaccine for COVID-19? That's a good question, and the people are working. If it is already in the market, probably another 3 months. So now it's June. Sometime November, December, if positively we can expect, we are not 100% sure. So we will see. We'll keep looking for it. But you guys be safe. More important.
Yeah.
Any other questions? One or two?
No.
Happy to answer. Okay.
Yeah, then I think we can wind it up. Thank you once again, Dharma, for your nice presentation.
Yeah, thank you very much, and I thank you all for the kind attendance. Yes, thank you, please.
Yeah, thank you. Tomorrow the session will start from 10 o'clock. Thank you once again for joining with us.
Yeah.
Thank you. Yeah.
Thank you.